Hello guys, my name is Brandon Martinez and I am performing a home wet lab experiment. It's based on raw and cooked potato versus hydrogen peroxide. Now before I start talking about the lab, let me give you guys basically some background knowledge and information. This lab is we're basically gonna be testing enzymes and reactions. So enzymes are organic catalysts, and catalysts are basically substances that help chemical reactions occur faster. They're not necessarily important for reactions to occur, however they do help. The reason being is that they require less energy for the activation to occur in a reaction. They're specific for particular reactions or a group of reactions. They're basically like Cinderella's glass shoe. You can't use one enzyme for one other thing. They're specific for things. Before I start talking about the catalyst enzyme, I want to give you guys that enzymes have two requirements for them to work. One of them being temperature and the other being pH. If the temperature is too low, the reaction is just going to occur very slowly because the enzymes they don't have enough energy to be reacting. And if the temperature is too high, the enzymes break down and the reaction of course won't occur. So the catalyst enzyme is found in eukaryotic cells, which is basically all living organic cells, and they help break down hydrogen peroxide. Below this, right here, this reaction that you're seeing is hydrogen peroxide reacting with the catalase enzyme, and it creates water and O2. My experiment's purpose is to compare and contrast the results of placing a raw potato and a cooked potato inside hydrogen peroxide. Remember that the enzyme catalase is found in all organic living cells, so potato has it. The materials that I'm going to be using today for this home wet lab is a potato, a knife or something to just cut the potato with, hydrogen peroxide, 2 glass cups or 2 transparent cups, it could be plastic, 1 fork, a pot, a stove, and I forgot to include water. So the procedures are as follows. First, you're going to turn the stove on and place water inside the pot, and then you're going to move the pot to where you turn the stove on. Second, you're going to slice the potato in half. You're going to place 1/2 inside the pot, and you're going to allow for that potato to cook until it's soft. How do you know it's going to be soft? Well, you're going to test it, and basically you're going to grab a fork and you're going to put it inside the potato, and if it passes very easily, then it's ready. If it still struggles a bit, then I suggest you let it sit for a couple more minutes. After it is cooked, you're going to remove that potato half from the pot. You're going to turn off the stove, and then you're going to place that half into its designated cup. Once that's done, you can place the other half into its designated cup. And then once both of them are ready, you're going to pour hydrogen peroxide into both cups, and then you're going to wait for the results. So here in this next slide, I have a video of me basically doing that procedure. And these were my results. So basically on the left I have the boiled half, and on the right I have the raw half. On the left we can see that it looks like I placed the potato inside water. There was actually no reaction. It stayed like this the whole entire time. However, to my right, it looks like I placed the potato into a Sprite basically. There's sizzling, there's bubbling over here. It's not as transparent or lucid. And you might be asking yourself, why is that? Well, remember that I mentioned that catalyst is required for hydrogen peroxide to occur, for hydrogen peroxide to have a reaction. And that's what's happening over here. The hydrogen peroxide is reacting with the catalyst found in the raw potato, and it's forming this O2, which is this bubbling right here, and the water, which is down here basically. Now You might ask yourself, well, why is this not happening over here? Well, the reason being is that we boiled the potato and the enzymes denatured and they broke down basically because the temperature over exceeded. So nothing is happening over here. Something would probably happen after like 10,000 years. Who knows? That's that. And an unknown not an unknown variable, but a chaotic variable that occurred, which I should have thought of. I was comparing my experiment with other people's experiments, and I noticed that when they placed a raw potato inside hydrogen peroxide, the reaction was almost instantaneous. However, for mine, it was kind of slow. And I thought about it, and the reason was that I placed this raw potato inside water while this boiled potato was boiling. And I'm pretty sure that once I took it out, I didn't allow it for it to dry, and I just placed it automatically into the hydrogen peroxide. And that's probably what's conflicting over here. That's why there's not as much sizzling as there should be. Yeah.